Hello, my name is Fidelia Asomani. I am a senior biology student at Notre Dame of Maryland University, and I am a Morrissey Honors Scholar. Today, I'll be presenting to you my research on discovering the truth about your yeast infection, the power of oxidative stress on WAR1 and Candida glabrata. So Candida glabrata is a haploid yeast commonly associated with superficial yeast infections. With antifungal resistance from mutations increasing, there has been more interest in the genome of yeast, such as C. glabrata. I focus on the transcription factor WAR1 and its role in other species of yeast is known, but in C. glabrata, it isn't as defined. In other species of yeast, such as C. albicans, which you normally will find in your gut, WAR1 controls large-scale phenotypic changes, and these changes have been seen to impact mating preferences, structure, and the health of C. albicans. So in this study, I will be studying the effect of the knockout WAR1, which I also refer to as delta WAR1, and its relation to oxidative stress, specifically the yeast defense against oxidative stress. You may be asking, what exactly is oxidative stress? And that is the imbalance of reactive oxygen species and detoxifying agents. What makes oxidative stress such a significant environmental stimulus is that oxidative stress is used as an immune response in many organisms. For example, it has been seen to activate apoptosis or cell death in S. cerevisiae, which is going to be your baker's yeast. To study this, in this study, what we're going to do is use reactive oxygen species, and the specific one that I'll be using is hydrogen peroxide. So what I'm going to do is add them to agar plates and then test both the wild type and the mutant or the WAR1 knockout, and in both of those conditions. And by comparing the growth of colonies in both conditions, I'll be able to see the effect of the hydrogen peroxide and oxidative stress on the health of the yeast. Now, previous studies have actually seen that 4 other transcription factors, those being YAP1, SKN7, MSN2, and MSN4, to control the oxidative stress response of Candida glabrata. And my hope for this study is that I will determine if WAR1 should be added onto that list. And ultimately, if oxidative stress can be used to address Candida glabrata and yeast infections. Now I would like to move on to my methods. And as you can see here, I outlined my methods here. So what I did was I began with Candida glabrata colonies. One of them was your wild type WAR1, and then I had 3 sister isolates of the mutant knockout WAR1, and they were grown on plates. Then I would take a colony from each plate and then grow it in an overnight culture. Then I would take the overnight culture and dilute it 250-fold. That diluted solution was then plated onto YPD and 4.5 millimolar hydrogen peroxide. And the control would be just YPD agar plates, and I would incubate both of the plates at 30 degrees for 48 hours. Now, to analyze the effect of oxidative stress on these yeast, what I would do is that after incubation, the colonies from each condition were counted. Then the number of colonies of each delta WAR1 isolate was divided by the wild type colonies to determine my percent survival. And if you look here, I will go into the results that I found from these analytical methods. Figure 1 is showing you the average percent survival of your Candida glabrata delta WAR1 and wild type WAR1 strain. What I found was that there was an overall trend that the mutant strains had a worse response to or a lower average percent survival than the wild type Candida glabrata. However, there were variations between trials. For example, if you look at trial 4 in the Delta War1 isolate number 3, it has a higher % survival than the wild type in that trial. However, the other trials do not show that same trend. So then I took the averages of all of those trials and then produced figure 1B, and the great variation between the trials is seen in the large standard deviation bars. And of course, having such a large standard deviation would make me not so confident in the results that I had. So my next step was to normalize my results. And that is what I got from figure 2. However, again, the overall trend was seen there that mutant strains of the Candida glabrata did not have such a better % survival than the wild type. However, again, if you look at trial 4 and isolate 3, it again had a higher normalized % survival than your wild type did. So I followed the same method again, and then took the averages of all of those, and again, produced large error bars. So in conclusion, our results did suggest that Delta War1 results in a decrease of Candida glabrata's ability to respond to oxidative stress. And we saw this through the trends of the % survival in your Delta War1 strains being less than the wild type. However, again, as I mentioned, the standard deviation was slightly concerning. So if I were to do this experiment again, I would perform more trials to increase my confidence in the accurate % survival for my wild type Candida glabrata. One possible reason for this change in the trend could be that in that last trial, the isolates were contaminated with the wild type strain. And one way to confirm or deny this would be to use gel electrophoresis. But I did want to point out that before performing our experiments, we did use gel electrophoresis to confirm that the isolates were what they were. And lastly, if I were to do more further study and give myself more confirmation, I would like to test or observe the response of Candida glabrata to oxidative stressors other than hydrogen peroxide. I think that would just give more information and better confirmation on if that War1 transcription factor was sensitive and controlled hydrogen peroxide. Thank you very much for listening. And I would just like to thank, before I end the Morrissey Honors Program, Dr. Zordin for giving me the opportunity to do this research and mentor me through the process. Our lab manager, Marcia Moore, for providing her assistance and resources. And lastly, the CFR&D for funding Dr. Zordin, which allowed this project to be possible. Thank you again.